All right, at this time, we are going into our student block, and I am really excited that we have two students presenting this morning. They have really given of their time to be here, um, joining in practice sessions, and they've made considerable efforts to share with us today. So thanks, guys. First up, we have Connor Hagmeyer. He's a master's student at North Dakota State University, studies under actually board member, Dr. Todd West, and he will be presenting on his propagation work with Betula Tianshanika. Welcome, Connor.
Thank you. All right. Hello, everyone. Today, I would like to really quickly share with you a little about my master's thesis project on the asexual propagation of Betula tianshanika. So Tianshan birch is native to Western China, and it's not well known in the United States. As I have been searching, I have not been able to find any information on propagation for this species aside from starting seed. The NDSU Woody Plant Improvement Program has made a selection from Tianshan birch seedlings and released the cultivar Emerdac, Emerald Flare Tianshan birch. This was primarily selected for its apparent drought tolerance, conical form, high quality foliage, and exfoliating white bark. Due to the genetic variation in seed production, this cultivar can only be propagated asexually. We decided to study the three main methods of asexual propagation that are used in the nursery industry today: micropropagation, also known as tissue culture, stem cuttings, and grafting. That way, when a nursery decides to grow Emerald Flare, we will have asexual propagation information ready to give them. So first off, in the micropropagation study, a factorial was developed that compared nutrient solutions, the cytokinin used, and the cytokinin concentration. The three nutrient salts used vary in concentration, with MS being high, LP is medium, WPM has a low concentration of nutrients. We chose to compare BA and zeatin as the cytokinin with concentrations of 0, 1, 2, and 4 micromolar to enhance shoot development and growth. And we measured any shoot that was over 1 centimeter. The propagule number determines the number of new plants produced from one explant and is a practical measure of overall growth. Adventitious shoots arise from single cells and can contain minor genetic variants, so they can no longer be considered clones, making them undesirable in large-scale tissue culture propagation. So after 6 weeks in our experiment, we found that growth was highest when zeatin was used as the cytokinin. We actually found that across all nutrient solutions, a zeatin concentration of 4 micromolar was the strongest performer and produced an average of 3.5 new propagules. Treatments with zeatin also averaged lower adventitious shoot production than BA, and explants performed equally well on LP and WPM, but explants on the MS media had much higher production of adventitious shoots. So the next stage of micropropagation then is rooting these explants. This can be done in vitro or ex vitro. Root production is stimulated by auxin, and IBA is most commonly used. After 6 weeks in our experiment, we saw, in the in vitro experiment, we saw increasing number of roots per plantlet, but there was little change in the overall root length. In ex vitro, roots were produced readily, with most of the control group rooting well. We also found that as the IBA concentrations increased, an increase in both root number and root length were observed when rooting ex vitro. So rooting ex vitro skips a step compared with in vitro rooting, making it the preferred rooting method in large-scale propagation. In addition to micropropagation, stem cutting propagation of Tianshan birch was also evaluated. We compared methods of auxin delivery, including powdered IBA dip, liquid NAA dip, and foliar IBA spray. The auxin concentration also varied between the delivery methods. The semi-hardwood cuttings rooted very poorly for us, and there was no detectable correlation between the treatments. And along with not producing roots well, the buds of the cuttings also desiccated rendering them useless. So much more research is needed before rooting gentian birch cuttings can be used on a commercial scale. We also evaluated grafting gentian birch. We studied 4 different rootstock species that were readily available, and we evaluated them for their short-term compatibility with gentian birch, and their annual growth was measured. We found that paper birch, Betula papyrifera, appeared to be the best candidate for gentian birch understock, as it had the highest percent survival at 58% and the highest growth rate at 85 centimeters. When grafted on river birch, the gentian birch appeared to inherit iron chlorosis, which I thought was pretty interesting because our greenhouse water is pretty high in pH. And you can see this in the picture on the bottom left with Betula nigra on the left and Betula papyrifera on the right. So in conclusion, we found that micropropagation would be the preferred method of propagating gentian birch at this time. Either WPM or LP nutrient salts can be used. And again, we found that 4 micromolar of zeatin resulted in the best explant growth. And then, although not necessary for rooting, 0.3% powdered IBA resulted in the best development of roots in our treatments. More research is needed for cutting propagation to be a viable option. And then gentian birch appears to be graft compatible with all the rootstocks that we tested, but Betula papyrifera did perform the best for us. So thank you all for listening, and I hope this information may be helpful to any nursery that decides to grow this plant in the future.
Awesome. Thanks, Connor. Couple questions for you really quick. Shelby, we have time? Yeah. Is there any evidence that you found, Connor, that there was a is there a relationship between papyrifera and the gentian birch? Are they the most closely phylogenetically related, or it just sort of happened?
So there's a lot of birch species. And as far as I could tell, the white barked birches are generally lumped in the same kind of clade, if you will. So we kind of expected those to perform better. It is fairly closely related to paper birch, but I wouldn't say we 100% expected that to be the best performer from the beginning.
Right. Liz wanted to mention that they have emerald flare at their lab, thousands of them sold for 2021.
That is great.
What grafting techniques did you employ here?
So we did a side veneer graft with just a single bud that appeared to work quite well for us.
And then last question we have here is, you mentioned semi-hardwoods. Was it availability of material? Were you able to get any real softwood material in your trials?
So when looking to find information on propagation of birch, I found that semi-hardwood was recommended. If I were to do this again, I would definitely compare with softwood cuttings as well. Also, we were dealing with only mature tissue, which I think we all know doesn't root quite as well as juvenile tissue. But yeah, that was simply because we didn't have access to enough plant material to do a large scale experiment with juvenile tissue.
Awesome. One more question. In terms of cultural conditions for this species, what are they like in the landscape?
So I don't have the fact sheet up here, but generally pretty easy to grow. Again, birch usually like it cool and moist. We are actually looking to trial this in other places around the country to see if the drought tolerance that we observed holds true in other parts of the country as well. Again, this was selected because it held its high quality leaves during a pretty severe drought we had where a lot of other birch defoliated. So that's kind of the primary reason this was selected. But overall, yeah, pretty conical growth form and really nice high quality foliage during the growing season, as well as a good fall color you can see in this picture here, and high quality exfoliating white bark during the winter season as well.
Excellent. Thanks, Connor.
Okay, awesome. Again, thank you, Connor. There were some comments in the chat box about rooting from softwood cuttings from Brian Maynard from out of the University of Rhode Island. So check that out if you're interested. And I forgot to mention Q&A for the students, so I'm glad everyone just picked up on that right away. Next, we welcome Josh Kite, a master's student at the University of Kentucky, where he is also the university's Nursery Crops Extension Associate. So no want of things to do in Kentucky. And we look forward to his presentation on research of foliar spray applications of KIBA in production. Thanks, Josh.
No problem. All right, can everybody see my screen? Let's see. All right, so like I said, oh, hold on. All right, there we go. All right, I'm Josh Kite from the University of Kentucky, and I'm going to talk to you about the impact of timing on leaf surface application of foliar auxins on rooting. So the background and the objective of this is applying auxin as a foliar spray has advantages over quick dip. The background is that, worker safety, application and efficiency. We have a reduced labor cost for the crop. The objectives is that we get to see how the auxins move through the leaf surfaces. And we also get to look at the timing and the sequencing of the foliar sprays. The first cuttings that we worked on was a timing and foliar auxin spray of KIBA on Viburnum dentatum. The control treatment was 2,000 parts per million of KBIA at a quick dip. And then we did a time of the foliar application at day 2 after sticking, 4 days, and after 6 days. And also into the treatment, we threw in multiple foliar sprays as well as day 2, and then at a day 4. And then we did another at day 2 and then at day 6 to see if there was going to be any difference with multiple sprays. So with the Viburnum dentatum, the experimental protocol was with the Viburnum dentatum. We did terminal softwood cuttings were rooted under a mist with bottom heat. Cuttings were treated with the KBIA as a foliar spray at different time intervals. Cuttings were then evaluated for percentage and the number of roots. And we had 6 treatments with 10 replications, and then we had 6 cuttings per replication, so 6 cuttings per treatment. What I'm showing here is a graph after we did stuff. On the y-axis, you have roots per cutting, and then we have the treatments. You have a quick dip, day 2 foliar application spray, a day 4 and a day 6, and then we had day 2 and day +4 or +6. The foliar spray was just as effective as the quick dip, and there was no difference in roots per cutting for the auxin applied up to 6 days after sticking. We did show with the multiple applications, though, that there was an increase of about 10 roots per cutting. Another graph here, we've also on the y-axis, roots per cutting, then the treatments, and then we also have rooting percentages that we've added to the graph at the bottom as well with the treatments. And then the conclusion of this is that we see that a singular foliar spray applied between 2 and 6 days after sticking did not impact the rooting, but there was an additive effect of multiple applications and increased roots per cutting by approximately 10 roots per cutting. We also did another plan in this. It was Euonymus elatus burning bush. We wanted to look at the auxin application of different leaf surfaces. And so the treatments we used was an Euonymus elatus, and it was an auxin location. We had a control of no auxin, and then we sprayed 5,000 parts per million of a KBI on a quick dip, We did an adaxile, adaxile with surfactant, a baxile, and then a baxile with surfactant as well. Each cutting contained a terminal bud of 4 leaves, and then the 2 nodes below the leaves had their pair of leaves removed and were just cut below the 4th node. And since the leaves were discotome leaves, so they had to be laid on their side, and either side in the 30 cuttings were sprayed with 10 mils of IBA solution. And then the statistics on it were 6 treatments at 5 reps with 6 cuttings per replication and 30 cuttings per treatment. Here we have another graph, and then on the y-axis, we got rootings per cutting. And then you see our treatments were no-oxin, quick dip, adaxile with surfactant, a baxile, and a baxile with surfactant. And there was really no difference in roots per cuttings between adaxile versus the abaxile application. But on the graph again, you know, we've got roots per cutting again, and then the treatments, and then we added rooting percentages. And if you notice on the adaxile and the adaxile with surfactant, you notice that we were able to recover some of the rooting percentages. It looks like the roots did a little bit better. And then here on this image right here, we just have obviously a microscope picture of a leaf, and you can see the top side, the upper surface, with all the dark green being the thick foliar cuticular wax, and then the bottom side of the leaf showing the stomates. So in conclusion on this, you know, we were able to find that there was little differences in rooting when the auxin was applied to the abaxile or adaxile surface of the leaves. But this also suggests that the main pathway for the auxin uptake was through the leaf surface rather than the stomatal openings, and that the cuticular wax may be a barrier to the auxin absorption, and so the surfactant may increase auxin efficacy. And so what we're going to look at in the future is like, does it work and where to go next, just to answer all the questions of how everything moves through the leaf, which will be starting in the spring.
Awesome, Josh. A few quick questions. In the first experiment with the Viburnum dentatum, did you see any rooting without hormone? Was there a control in that?
No, we did not do a control. We did not do a control with no rooting without hormone.
Gotcha. What was the concentration in that first experiment? It was 5,000 in the second.
Yeah, sorry, I forgot to mention that. It was 2,000 parts per million.
Gotcha. I don't see any other questions. Yeah, that was excellent. Thank you, Josh.
All right, no problem. Thank you all.